Hi, good evening, sir.
Yeah. So how many molecules you got in your assignment? Just now I'm checking. Assignment PPTX, right?
I got 8 molecules, and then remaining 2, 3 I just showed in the PPT that is having very less resolution, 1 point something, but it is not coming in our range. That's semi domain.
Okay, resolution is 1 point something.
Yeah,
1 point is a good resolution.
yeah, it's a good resolution, but it is not coming in our semi domain region.
Oh, fine, 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 fine. Okay. Okay, if it's the case, we can go for a modeling. I'll try that. But however, I'll teach you all the techniques, how to proceed further.
Okay, sir.
Yeah. Rather, I am presenting. Can you present the data now? What about your Ubuntu? It got installed?
Yeah,
WSM.
yesterday, not from the Microsoft Store, I couldn't download it. Simply from the Google search, I just wrote Ubuntu and downloaded. That's fine. Or you had any specification?
No, ma. Like, can you find the icon WSM? Have you took your help of your husband, like to install? Like I told,
Yeah,
na.
he.
Like,
Can I share the screen? I will show you
please,
where
please,
I
please.
have.
Yes, yes. Yeah.
Yeah, this is what I have got obtained. Like, can you see in softwares one
Oh
Ubuntu?
my God. Actually, you got an ISO file. That's to install uh, dual OS. It is like installing
Okay,
OS. But I want
okay,
you to use in command prompt of your Windows.
okay,
I have shared you a
okay,
link. You got the idea, na? Actually, using that, people will do copy that in a pen drive, then they will insert in the pen drive, and they will boot for dual OS.
okay.
What do I say? Like, we need a WS. Does he have a laptop now? He's with you. He can try in his
No,
laptop.
now he's not here. Uh,
Do you have any updated system now in your hands, like anywhere?
he's having his company laptop. I don't know whether he can do something in that.
If
I'm
anything,
not sure.
any other system is there, can you keep it side so that we can install the same
No, no, right now, no, sir. I don't have because he's
any
not
system?
here.
Okay, fine. Right, we can move on to next step. We'll come back to this Bromax stage. When we have doing Bromax, definitely we need this. Or
Okay, sir.
I can do one thing. I can run your data here, and I'll show you. You can see the recording how I perform
Mhm.
that. Once you install, you can do on hands-on. Is that fine for you?
Okay, okay, sir. Like, should I ask my when my husband comes? Can you
Yeah,
like
you can
any
ask him to
rules?
join here. If he is there, you can ask him to join even. Like this is not the time he arrives. It will take time for him.
I have to ask him, sir. Just
Okay, fine. Or we can plan on weekends someday, not Sunday, but Saturday. Right, we'll sort it out. Rather,
okay, sir.
now we'll go for confirming the molecules.
Okay, sir.
Can you please share your info?
Mhm. The presentation which I have
Yes,
sent.
yes, yes, yes.
So you can see.
Yes,
Okay. So this
yes.
is first 4R1V. Again, this is not in our region. Amino acid 1061 to 1341 it is.
So our
It is
range is
having
our range is
25 to 517.
okay, not totally in the range. So no need to
No.
take. We can avoid.
Okay.
But a good structure it is. Yes, this one.
Yeah, this is like that. And then next one is 5 LSP. This I found in one paper
Hmm.
regarding the allosteric binding.
Okay, fine, fine.
Okay.
They even mentioning as hepatocyte growth factor receptor 231 sequence. Can you see the
Yeah.
range?
Yeah. Then range once I have noted it somewhere, sir. I will just.
Yes, yes, yes.
Okay, I read that paper. Somewhere it is there, but in my previous presentation also it is there, sir.
Oh, same
But it
protein,
is not. So
not
same
using.
protein I have taken for docking 5 LSP.
Okay,
But
fine.
okay, okay. Then it is coming in that range of our protein.
Hmm.
Okay.
5 LSP.
Um,
5 LSP.
yeah, range of semi-domain. I think. Let me see. In my previous presentation also it is there.
Okay, fine, great. So 5 LSP of or I'll teach you one more thing. Can we perform that in the meantime? I'll see a few more, and if it is not
Okay.
working, cross check also. Fine.
Okay.
I'll teach you PDB blast. Right,
Okay.
we'll go further. So as of now, you can make a note of 5 LSP. So it is having oh, not in the range of semo domain. You have mentioned that
Ah, yeah.
not in the
But I'm not sure now. I want to check that range
cross
which
check.
I have got. I want
Okay,
to
it's
confirm it
230.
once more
Yeah, 0 to 220 it is written.
because I remember that it was in that range because I selected that because of that. I think.
Okay.
I don't remember now. I have to confirm it, sir.
It is
Okay,
written
then
as
this one is.
repeat. It is written as
Huh?
it's in repeat. Just a minute. I'm showing one thing.
Okay,
Um,
I'll stop sharing.
no, no, you proceed. I am simultaneously checking your structures. I'll show again.
Okay.
You can
Oh,
complete this. Then I'll come back. Yes,
okay.
ma, please.
Sir,
Yes.
in the same way in that it was written like chain A and what is chain D? It is written, right, sir?
Yeah, yeah.
So two chain is hepatocyte growth factor, but I couldn't find that in the PyMol.
No, actually, they might have referred us. Author referred as P, and they given the designation as D.
What is the meaning of that? I didn't understand.
Wait a minute. I'll share my screen. Let your screen be shared. I will overshare it. Okay.
Okay.
How about now? Can you see my screen?
Not yet.
This one, na 5 LSP.
Uh, Wait, sir. I cannot see your screen.
Okay.
Okay.
Same thing, right? 5 LSP.
Yes, sir.
Fine. So in this, if you observe, and they have given
Mhm.
two chains, A and D,
Yeah.
but author have mentioned as P. So we
Okay.
have to go. We have to go for this A and D only. A and D. So these A and D are unique sequence. There is no doubt in that.
Okay.
Okay, and
But it will not be mentioned.
it will be mentioned. If you open in Primal, you can see that.
Okay. D also will be there.
D also will be there. Let me show. Have you tried everything in Primal while you're retrieving the structures? Have you checked the chains?
Mm-hmm.
So I'm opening now. Let me confirm. So I'm putting how to find chain identifiers? Displayed sequence mode,
Sequence code chain
chain
identifiers.
identifier.
Okay.
Okay, fine. Now they have given us HLPSTXY, correct?
Yes.
Now it clearly means they are going with author's ID. So author have considered this as P.
Okay.
This chain is P.
Okay.
So
Okay.
we have to go to P.
Okay. If you want that. Okay.
This is the chain what they mentioned.
Mm-hmm.
Okay, yesterday I taught you to delete everything and save the remaining, correct?
Yeah.
Today's class I will teach you how to save only the required file.
Okay.
A, a small modification. Just I have to click select action. I have chosen P. Instead of removing everything, I'm just renaming with 5LSP underscore P. Clear? This I
Okay.
rename. Now what I have to do, I go to how to save the file generally. File,
File.
export
Export
molecule.
molecule.
I might have advised
Then.
you to save as it is, correct?
Yeah.
Now I'm not removing anything, only I've selected the required molecule. Now I'll go to 5-LSP hyphen P.
Okay. And then
Now
save.
save. Just save. So only one option you have to make is you have to convert back into PDB.
Mm-hmm.
Then I'm putting in your manuscript like, yeah, 5-LSP hyphen P. Now I'll close
Okay.
all these things. I'll go back to your folder.
Mm-hmm.
If I open that, I'll have retain with only
That
one,
region. Okay.
that region only. Now I'll go here. Okay. Look at the range. They're saying 519 to
Mm. That
1736. But
okay.
I have a confusion now whether this region and that region is matches. I'll cross
No.
check.
Okay.
We have to check the sequence. Might be they might have changed, I do not know. We have to check the actual sequence. I removed it. So now we are left with only the protein structure.
Mm. Okay.
Fine. So this is the way to save. Now you can go back and say the export molecule only what is left out. Save as it is.
Okay.
Can you understand what I did now?
Yeah, you deleted that molecule, right?
Molecule, and again I
The ligand
resaved it.
molecule.
Yeah.
Yeah.
While I'm resaving it, I just now I gave as it is. If I have to save, I have to save enabled as it is.
Okay. Otherwise, you have to selection. From the selection, you have
Select.
to select what you
Yes.
renamed.
What I need, that only I'll save. If not, I
Mm.
remove whatever is not required and save as it is.
Okay, so.
So this is done. Okay, Nemy. So this is done.
Mm.
We are ready with the structure. But now we have a doubt whether this one and that one are same. Correct?
Yeah, the region. Yeah.
Region are same. So okay, let us have this 5-LSP in the count. Can we move on? Okay, I'll share my
Yes.
screen. Uh, Okay, how about this is good. 1.73 is a good resolution. How do you
Um.
say it's not a semi domain? Again, the range is
Oh,
different.
this actually is not semi-domain because the range is different. Yeah. And
73,
also here
73. Yes, yes, please.
7V3R. And this one actually checked in the alpha fold what you showed, sir.
Hmm. Yeah.
In that when we go, we can see that proteins. Along with that proteins, we can see the range also, where which
Okay.
they have highlighted the range where it is.
Okay.
Okay, so
So
that like that, I most of the
from here,
proteins are
I can
not
understand it's 11 to 319.
from
So
11 to
319. Can you see here what I'm showing you?
sir. Wait, 1 second. Okay,
Uh, See, I have to
it is
hover the. I have to keep my mouse over this bar.
okay.
If I keep over the bar, can you see the position over here? So I mentioned here, like, hope it should visible now. Correct? 319.
Okay, 319.
So what's the range we
11.
require?
Oh, mine is from 25 to 500 and 15, 510 like that. It is.
Okay, we need 515.
Mm.
Now, if I go down, it is written as okay, threonine kinase. This is the location. Can you see the domain? PFAM refers to the protein family.
Sir, where it is written? I can't find.
Can you see the see whenever I keep the mouse, you can see the
Mm-hmm.
highlight of it. It's called hovering. Like when I
On the
keep
top,
mouse,
protein
yeah,
tyrosine and
yes, yes. Can you see that?
yeah.
They are saying that by 50 to 310. Wait, I will just browse through. 50 to 310 refers to this domain, so and so domain. The domain is serine threonine kinase. Correct?
Mm-hmm. Okay.
This is what we require, right?
No, we need more of amino acids. It's not till 310. It's coming under
We need till
500.
500.
Mm. Yeah.
Oh, we need till 500. Again, right, let me have. Okay, have a tick mark. The 7-3R. 7V3R,
Okay.
we have a tick. Okay, CMAT in a complex. We'll come back. Right. Then next one.
It's 5 EYC.
Okay. 5EYC.
Yeah.
5EYC. Suddenly it became slow. Okay, let it load. Meanwhile, what it is? Like serine, threonine, protease kinase, catalytic domain.
Mm-hmm.
But I have wonder,
Yeah.
like, sema domain is a part of, sorry, tyrosine kinase is a part of sema domain.
No.
Okay, these are the domains. Like, how come then they are describing the tyrosine domains? One
Tyrosine
second.
kinase is a not present external domain, right? It is intracellularly present, no, sir?
Yeah. Yeah, the first one what we have seen, correct. High resolution missing amino acids, high resolution. Oh, 6 is too bad. 2.8. Why you rejected this? It's only HGF, not receptor, correct?
It is not receptor.
Okay. So thing is, you should mention this as a low resolution.
Mm-hmm.
Higher the number,
Okay.
lower the resolution. Okay, hereafter you have to
Okay.
mention
Okay.
it. Lower the resolution. Okay. 25 to 567, but in time mode, when we open, it starts from 495. I think this will work, I guess. 403U. Again, 3 angstrom is not good. 2.99 angstrom. So let me check. Okay, they are very clearly saying it's a crystal structure of C-metal complex, correct?
Mm. Yes.
309. Form hepatocyte growth factor receptor. They are very clear. So this is the range they are mentioning.
Mm-hmm.
So they have the range of 1 to 304.
Okay.
They are not stopped till 500. But in
Mm.
UniProt, okay, fine. Let me see. So all the proteins they refer to same UniProt called 508581.
Yeah.
So let us go and find in that 508581. So see, can you see the structural summary, Ms. Nemy? Like
Sir, have you
overall,
went there in
yeah,
this?
I just click over the domain. There is a domain. Can you see this domain? I clicked.
Okay.
When I click, everything is interlinked. Or I have either you can click that or open this.
Okay.
When you open this, they are showing the associated proteins for this particular receptor. There are these many associated proteins.
Yeah. Okay.
See here, everything is partial. Can you see? All these PDB IDs are partial.
Okay. The gap what we are seeing because of that.
Yeah, that's why. See, this is the complete sequences. Only this has the complete sequences.
Okay.
7M09. Is that in our list? Not in the hit list?
No.
Yeah, the 7M09 is saying we are having the entire sequence, like 1390 is the entire
Oh,
protein.
okay.
Yeah, 728. Great. It's not X-ray, I guess.
Electron microscope. Yeah.
Fantastic. Can you see EM where you are found?
Yeah. The
Correct.
method resolution 4.
Four. Fine. EM, they use the EM.
Okay. For EM, how much resolution we can? It's permissible.
See, again, EM is better than X-ray. I don't know the
Okay.
limit, but generally for X-ray, we will stop there. Okay, 7M09. Fantastic. This is having the entire structure. We'll
Okay.
check the
Okay.
quality. We'll check the quality. Is there any missing residues? We will see. Actually, crystallography is the classical method. So
Mm-hmm.
people, while they doing it, a lot of drawbacks were there. But
Okay.
wherever you kind of EM is a recent method,
Okay.
so we will prefer this. Yeah, some missing residues are there. It starts with 34
Mm-hmm.
and we have till 500. Okay. No, they have used lot of linkers now.
Okay.
386, 466, P chain, F chain. Yeah, a lot of linkers are there. Let us keep in wait list, not to confirm. New protein. See, if nothing is working out, no go, we can go and model it.
Okay.
So, can you see the number of domains? When I keep the browser, they're saying 40 to 124 is called pan domain.
Mm.
Pringle, okay, pfm, trypsin domain. So our range is, let it be. We'll come back to this. Okay, that is not working out. There is another structure called 7M08.
Mm. Okay. But
7M07. I
that
think,
on that too there is gap, sir. So we are checking whether in our
mm,
region if it is complete or not. That is
yeah,
what we are
yeah,
checking.
yeah, correct. So, again, here it is not covering. We don't bother about that. We need
Mm-hmm.
from 0 to 500. Am I right?
Yeah. Yeah.
This is the domain.
Mm-hmm.
This domain we need.
Yes.
So whatever covering in this domain, we can go for.
Mm-hmm.
Something is called, again, some gaps are there. 7WVZ, 6WVZ. One thing is very obvious. None of the structures are having complete residues. Correct?
Yeah.
Yeah, it is again 3.10. So no need to waste time. Rather,
Mm.
we will go for existing methods. Can we move on?
Yeah.
Good. So there is a thing called this model workspace. I'll share the manual now. You can work simultaneously along with me.
Okay, so
Yeah. So please type this model workspace. I put in chat.
okay.
Meantime, in your free hours, kindly check with your husband how to install WSL in Windows.
Okay.
Yesterday I shared you a link, right? Do you save the link? No, it disappeared.
No, sir. Yeah, it disappeared, I think, sir.
Or I'll
I
put the
don't
link.
know how to save it. Here in this, we can save. I don't know how to save.
Oh, do one thing. Whatever I'm sharing in Google Chat, kindly paste in your some notepad or somewhere.
Okay.
If I'm putting this link, like this is works, is you forgive this. Like whenever I put some link, kindly take that and save somewhere.
In the notepad, I will save, sir.
Notepad, yes,
Sys model interactive workspace.
correct.
Okay.
Okay. This link is to install WSL. Whatever I'm sharing now, please save somewhere. Or there is another option. Go here, bookmark. When you open the link, kindly open the link,
Okay, sir. I will just open. Okay.
and there is an option called Control D. Please press Control D in your browser.
Yeah, control D. Okay, bookmark.
Yeah, you can
Done.
bookmark this. Whenever you want, you can go to this bookmark. To assess the bookmark, there is an option. You can click some open. What browser you are using, Nemy? Like any browser, you will see some three dots at the end. Can you see the three dots?
Yes, yes, yes.
In that, you can go. There will be a column called bookmarks.
Yes.
In that bookmark, you can find the recent bookmark what we have made today.
Yes.
Yeah. So whenever required, kindly open this and show him. He will help you.
Okay.
Oh,
Thank you, sir.
okay, this should be ready before the next class, and you also have to practice all the small things what I gave in the previous workshop,
Okay.
yes, previous session. Now let us go into Swiss model workspace. Okay, what do
Okay.
you mean by okay, before going there, what do you mean by modeling? Why we need to model some structures?
Like in my case, if there are no protein available in the PDB or in the databases, we have to model.
Correct. We have to model. Correct. What is the
Yes.
benefit of modeling then? Like we have to model. Moreover,
Mm-hmm.
it is enough today's world now. It will go for multi-template modeling. It will go to a column called multi-template
Modeling.
model.
Multi-template modeling. Okay.
Yes. So multiple templates it will take. And today's era, we also will use another thing called A AlphaFold.
Mm-hmm.
So AlphaFold is more effective than anything else.
Okay.
Yes. Hope my screen should be visible now. Is it visible? No?
No, sir.
Yeah. How about now? Still is pending. Let me refresh.
No, sir. I cannot see. Okay. Now it is visible.
Fine, fine, fine. So we are going to see homology modeling using Swiss modeler.
Okay.
Okay. Generally, to understand, it's enough to have an amino acid sequence.
Mm-hmm.
Okay. Amino acid sequence is called target, and the experimentally resolved structures are called templates.
Okay.
So what we are going to do is we are going to use a target. We have a target sequence. So what is
Mm-hmm.
our target sequence here?
Um,
Semen.
25, 20, only that domain we have to say?
Yeah, domain we have to say, like, that is the sequence we record, correct?
Yeah.
So that is our target sequence. Then what is the template structures?
What is available in the
Yes, what are
database?
the PDB experimental structures are available, those are called template structures.
Mm-hmm.
Okay, for example, you might know idli, you might know cupcakes, correct? What is the purpose of cupcakes?
Sweet.
How yeah, they are sweet. Okay, how they made the shape? It is based
There are
upon the
molds.
it is based upon
Mm.
the container, correct? They
Container.
will have a container, and they might pour the dough, and they will allow to keep in microwave oven, then they will acquire the shape. Am I right?
Yes.
Same way, the container here is our template structures. So we will have the
Mm-hmm.
template structures. Okay. Now our sequence will try to match the template structures.
Mm-hmm.
When our sequence is trying to match the template structures, they will orient according to the shape.
Mm-hmm.
By this way, we will achieve the model structures.
Okay.
So classically, they will say we have to check the identity. We have to check the
Identity.
identity. Okay. Uh, Before this Swiss model workspace came into an active state, we have to go with range wise. We have to use multiple tools. One of the important tool is called Blast. Have you heard of Blast?
Okay. Yeah.
What is Blast?
We get sequence from that.
Yes, that is called Basic Local Alignment Search Tool. It's a search engine.
Okay.
Uh, I'll share a browser now. So there is a thing called Blast. basic local alignment search tool.
Okay, should I take this in mind and bookmark it or?
No, you can perform. You can write
Okay.
somewhere. Just PLAST. You can again. You can have a book. Or you can have a paper and pen with you.
Ah, yes, it is there with me.
It is that. Please make a note of this. Or you can also see the recording if you want. If you can go back and
Oh.
see the recording. Are they
Mm-hmm.
providing things, neighbor man?
Yes, yes, yes.
Yeah, you can see that. These are not included in the manuscript. These are extra points, like what is happening behind. I'm trying to explain you so that you can
Okay.
also explain in future. There is a shortcut. This shortcut we can do in 2 minutes. No
Mm-hmm.
issues. But I want to explain what is going behind.
Okay.
Right. So Swiss model workspace. I hope it should be visible. Can you see different
Yes.
types of blast?
Yes.
Nucleotide blast, protein blast, blast X, T blast N. Okay.
Okay.
So if I have a nucleotide sequence and I want to know how many nucleotides are existing in the nature or already people have identified. For example,
Mm-hmm.
you are an enthusiast. You're going for a scuba diving in some Spain and collecting a sample from it. Okay, you came to Andaman Nicobar, you collect some samples, and you go back to your lab and try to know what that sample it is. Okay, the fungus or not sepal. Do you want to characterize from the startings, from the scratch? Is it required?
No, it will be there in the database.
It will be there in the database. You have to find only the sequence of the structure, sequence of that
Mm-hmm.
particular organism. Okay, you can go for 16s rRNA sequencing or whole genome sequencing. Just by having the sequence, you can put in the database and try to find the character. Correct.
Mm-hmm.
The same way, we have n number of sequences in the database. Same like protein. In protein
Mm-hmm.
blast, there is an option called PDB blast. There is an option called
Okay.
PDB blast. Fine.
Okay.
So PDB blast, for example, we need a domain from 25 to 500. Am I right, Nemy?
Yes.
So we are going back to UniProt. So UniProt is the primary database. There are two types of databases: primary database, second derived databases. So primary are something
Okay.
is having a direct connection with experimental biologist.
Okay.
So they are saying, what is UniProt? UniProt is the world's highest, leading, high quality, comprehensive, freely accessible resource of protein sequences.
Okay.
So nearly 5 lakh sequences are there in SysProt and 2 crores or 2 millions of sequences in Crumble associated database. But these are unreviewed. But we'll go for
Okay.
reviewed one. Like we are going for highly reviewed. Like people have done annotations, adding up
Mm-hmm.
information. So let us give the type name as hepatocyte growth factor. I'm okay with the name. Is that fine? Is
See,
it correct?
CMET. Okay.
Hepatocyte
Receptor.
growth factor is a receptor. Okay. Hepatocyte growth factor receptor. Correct.
Yeah.
So it is showing nearly 60,902 results.
Mm-hmm.
But I'll go with highly relevant one and I'll go with humus Homo sapiens
Mm-hmm.
at GF. So P14280 is the same number they refer to. No?
No, sir. No.
What
I
P?
think we have to search CMET.
Okay, and let me check CMET. C hyphen met. Is that fine?
Yeah. Yeah. Yeah.
Okay. So hepatocyte growth factor human. Okay,
Receptor.
whether this number is matching with the PDB IDs P08581 in your records.
I don't have it now, sir. I think yes,
Okay, do
because
one
it is
thing. Can you say any one of the PDB ID from your PPT?
from my PPT 1SH5.
One one HS5, correct?
SHY. SHY. Okay, I'll type it.
One HSY, correct?
Yeah.
This one?
SHY. No, sir. I
Okay.
have put in the chat.
Yeah. I got it. One SHY, correct?
Yeah.
What I want to cross check is whether my input is correct. So again, let me go to, okay, they are saying P14210.
Yeah.
It is not the same.
Mm.
But your recent PDBs I can see, like four, just now you shown me, no? Like 5LSP.
Yeah.
Let me check 5LSP. Yeah, they are referring to P08581. Same thing.
Okay.
Let us go with this. Is that fine for you?
Yeah.
Okay. So we are going for P0858. Here, I have to go directly into structure, or there is a thing called sequence. Where do you find the sequence?
On the left hand side, sequence and isoform it is written. We can directly go from there.
Yeah, we can go directly. Can you see the sequences?
Yes.
You mean, okay, then you mean here,
Yeah.
sequence of isoforms. Fine. Yeah, we are going directly sequence. In that again, multiple regions were there. Correct? We should not get
Yeah.
confused. Like if I click here, the entire sequence will come.
Mm-hmm.
But I want only, I don't want the entire 1390. Rather, I need a part of it. Am I right?
Yeah.
So since we need a part of it, we have to go for the domain wise things. So I go for family and domains.
Okay.
So SEMA domain is none other than 27 to 515.
550.
So you can make a note of it. I just have to click this.
Okay.
This is the exact domain I need, correct?
Yes.
So without further any loss or anything, I just copy the sequence
Mm-hmm.
to blast and paste it. Clear?
Okay, so.
Then I will go for I have which database I have to search for. So if I'm choosing NR non-redundant, it will search for another sequence database, protein versus protein.
Okay.
Rather, I'm going for PDB. The sequence I have to search again protein data bank,
Okay.
and I have to confirm with the organisms. Correct?
Yeah.
It is optional, but anyhow, I'm giving. I don't want to confuse, but if you want, you can give. Let me check all the
Okay.
results first. Then I'll clicking in a thing called blast.
Mm-hmm.
Just two options have changed. Am I right? What are the two options? Just I change the PDB and given the
You
sequence.
pasted the structure
Correct. This is called
sequence.
target sequence, and this is called searching against the database.
Okay.
Now I got a list of values.
Okay. On the below you have.
Yeah, I have list of alignments. To make it simple, I'm going for graphic summary. So 22 hits were there.
Okay.
In the 22 hits, to know the description, check, you can see here, they're saying the domains. SEMA domain, SEMA met, correct? SEMA super family.
Mm-hmm.
And from 100 to 450, it is called HGF binding site, INIB binding site. These are called descriptions, family, super families. Now, to understand, I'm going for descriptions. In the
Mm.
description, what you can find? You have to find for three factors. One is called query coverage.
Okay, so let me note.
What is a query coverage? Zero,
Um.
100% is query coverage. Query coverage refers to if I have to compare two humans, okay, I have to compare the entire body of them. Am I right?
Yes.
I should not compare only their hand. Both their hands should not compare. Correct.
Yes.
Same way, if I have to compare two sequences, you can also see the both sides. Just a minute, I will show this. Can you see? This is called
Yes.
pairwise alignment. This is called pairwise alignment. In this pairwise alignment,
Pairwise.
pairwise
Pairwise.
alignment.
Okay.
What do you mean by pairwise? Means I'm comparing pair. These are bioinformatics terms. No need to confuse too much. But what you can understand is 100% they are matching.
Okay.
Not even a single change is there. Do you know what sequence it is? Again, 7M07B.
Okay.
I think we have
That is
seen
what we
only
checked here. Uh,
correct. Just few minutes back we have checked 7M0B. But unfortunately, it is having some missing residues.
yeah.
Okay, we need only 515. Okay, let me check. But we will let us prefer this as a model template. What we can do is we can prefer this as a template. That's what we can do. And the second thing again, look at here. The second query is what? Which is matching 100% again.
Mm-hmm.
7GCU. We have not checked this. Let me check.
6 GCU.
6GCU. If it is below 2, we can go with this without any doubts.
6
I got
it is. I think it is there in the PowerPoint
Yeah, it is
presentation.
there in the PowerPoint. But have you found any missing residues?
No, since it is 6, I didn't open, sir,
You not even open. Huh? Let me check.
because it is having 6 resolution.
Yeah, it is poor only. Definitely they will have some drawbacks. Let me check. Some reason might be their long entire chains are there. Na? Might be a reason they had a very poor resolution. But let me check. See, can you see the fragments?
Sir, I cannot see your screen.
Oh, sorry, just a minute. How about now?
Yes.
So this is what I have opened. Okay, here, see, can you see the gaps?
Yes.
Right, 40 we started, but again gaps are there. From 301 to 311 is disappearing. 376
Mm-hmm.
is disappearing. Lot of gaps are there. So again, if you work on it, we may lose some sequences. So it's not ideal to take. My dear friend,
Okay.
the thing is, many people won't teach you this particular specific point. We
Yes.
unnoticedly will go and take sequences. When you submit for publications, they will ask questions. Why you have chosen this? How can you prove this? And might be some important residues might be there that you not reported. Correct?
Okay.
That will be an issue. That's the reason we are worrying too much. Now you are clear. So we can't go
Yes.
for all. Rather, what we have to go now? The only point is we have to go for modeling. Can we
Mm-hmm.
proceed?
Yes.
So we cross check it. So to have modeling, the point is copy the sequence,
Okay.
go to Swiss model workspace, Swiss model workspace. See, there are Swiss model workspace have 3 different points. Okay.
Okay.
Sorry, I'm not okay. I went. Okay. So this is again developed by Switzerland Protein Consortium. Can you see? Swiss Institute of Bioinformatics.
Mm.
They're very classical people. Like they are very part from Human Genome Project. Okay. There are three
Mm-hmm.
methods. Can you see at the side?
Okay.
One is whether
Sequence.
we have to give only the sequence or we have to give target template alignment file. Target template alignment file. Or
What is that, sir? That is same as that of sequence itself?
yeah, here we have to give only sequence and just look at. We no need to bother
Mm.
about anything. It themselves will choose and give the best result.
Okay.
In our case, it will choose the first one. What is the first one? What is the best hit here? So where is the blast?
What we saw, okay, 7 MOB,
It
MO7B.
automatically will select this because it is
Mm-hmm.
having high relevance. But
Okay.
for some other reason, if you're choosing this 403T or something, it is user defined. Then I have to give the alignment file of it. I've
Okay.
shown you the alignment file now, just now, like this one. Can you see the alignment file? Here, this file.
Ah, okay.
I can save this alignment separately.
Ah, okay.
But this is not our intention. I don't want to waste time here. These are very basics.
Okay.
Can you see here download?
Yeah, yeah, yeah.
This is called alignment file.
Text aligns.
Yeah, this ALN you can go and upload inside.
Ah, okay.
But that is not required now. Rather, we'll
Okay.
go and directly paste the sequence. We'll go for advanced methods, like it is automated. We'll go for
Okay.
automated method. So one thing you have to remember: don't go randomly, go domain wise. Okay, I'm clicking the family domains. Then I found the particular domain of whatever I'm performing you now. You can later on looking at the recorded video, you can practice.
Okay. Yes,
Okay,
sir.
now like copy this, go to Swiss model workspace, go for the basic version directly. Support as inputs are sequences. I'm pasting the sequence.
Okay.
Once I pasted the sequence, can you find they themselves started aligning it?
Mm-hmm.
Now I am defining a name. This is called sema domain. I CMAT sema domain. It's up to you. If you want, you can have. If not, no worries. Okay. You also can give your mail ID. Okay, your wish. Mail ID you can give or directly you can download the structure. Right?
Mm-hmm.
Now there are two options. Just I click build model and wait for the results. It will go till the end.
Mm-hmm.
Or I can search for template and go step by step.
Okay.
As of now, I'm going to build model. Later on, you can explore all the options.
Okay.
I'm going directly to build model. So auto model is running. More models are still to be built for this project. Can you do the same now? It's easy, right?
Yes.
Can you do the same now?
Yeah.
So I'll keep it loading. Meantime, you can open your browser and
Mm-hmm.
start doing it simultaneously. Is there any doubts so that I can help?
No, sir. No, sir. I have done the same way I have done
Good.
now. Meeting.
These are completely new for you, or you heard of it before? Like we have to
So
do a
this
blast.
model I have. Yeah, I have heard about it, sir. Swiss model.
Hmm.
How you are the same procedure I know.
But the background of below it, behind it is
Hmm.
they are doing a blast actually.
Mm-hmm.
This is the way we have to find the defined user defined sequences. Like we have to go for a blast, classically take that alignment and go step by step. But I
Mm-hmm.
want to make you simple and effective. This is a simple and effective way. Now
Okay.
we you can see entire thing is done. Our output is ready. We can directly download the files and proceed the interaction. But I just spent 2 minutes, couple of minutes to explain what happened here. Look at here.
Okay.
So I told na by default what selection will be done,
Yeah.
the best one. So totally 9 templates were with good results. In that they preferred for 7M09, correct?
Yeah.
Then see, and then they look for stoichiometry, they look for topology, everywhere, and they also found the sequence similarity, and they look for alignment, everything they made. Okay. Then they finalized with 7M01, and we got a model.
Mm-hmm.
Again, there are several models are there for this. We prefer for the first best model called 7M01, this model based upon this.
Mm-hmm. Mm-hmm.
And what is this? Can I see here?
I don't know, sir.
Things you
It is
can use
written
for-
Ramachan.
Correct. These things you can use for your presentations.
Mm-hmm.
Your model structures are that defined. You can do the same and click the Ramachandran plot. You can create a document. When you keep on creating document, write about it, it will become a manuscript, right? Can you see that, Ms. Nemy? Like this is the way. Have you heard of 5-side plot, Ramachandran plot?
I have heard it, but I don't know what is it.
Okay, simple. Any protein will have the dihedral angles. Any protein will have
Dihedral angles.
dihedral angles. See, actually, proteins. Okay, what proteins are made up of?
Amino acids.
Okay, amino acids. They are not flexible actually. Amino acids
Okay.
are flexible only in particular regions. Okay, this
Okay.
may be a better picture. Can you see the big picture? For example, I am taking this alpha helix. This
Mm-hmm.
particular region, two amino acids are there, correct?
Yeah.
So the two amino acids are made up of, for example, they have a carbon, oxygen. Hope you are clear with basic chemistry, like chemistry of amino
Yeah.
acids, correct?
Mm-hmm.
So entire protein won't twist. For
Okay.
example, this is an amino acid. Can you see my fingers?
Yes.
These are two amino acids, and these are carbon, hydrogen, nitrogen, oxygen. I can't bend like this. Rather, how will I bend? There are two options for me to bend. Pi, psi.
Okay.
Can
The
you see the phi chord? Yeah, they act as a plane. That's the point here. They act as a plane.
plane.
So all together only I can bend. This is called
Mm.
stereochemistry. This is called
Stereochemistry.
stereochemistry. You can later read about these words. What is called steric hindrance,
Mm-hmm.
steric effects. So any steric effects are due to the intermolecular clash. This is not actually what we see in the thing. Okay, so that's the reason we'll go for Ramachandran plot. How far these 5-side plots are fixing these angles. So whatever the mark they made here, these marks are none other than best. These marks are none other than the suitable regions, allowed regions.
Okay.
So
Allowed regions. The movement allowed regions.
yeah, if they fix inside the box, the particular graph, they made some contours, right? When they fix inside here, we are saying it is an allowed regions. It's a minimized structure, good structure.
Mm-hmm. In that region, the amino acids can move. That is the meaning.
Yeah, and they are fitting. They are fitting in this box. Can you see the green box? There are more allowed
Yeah.
regions are there.
Mm-hmm.
If you click here, you can get the screenshot of it, and later you can
Mm-hmm.
analyze. So if anything is lying in the white color regions, then it is called disallowed regions. Fine,
Disallowed.
disallowed. So fortunately, our all the peptides are, so all the amino acids are inside the allowed regions. and favorable regions.
Mm-hmm.
So take
Okay,
my
sir.
later on you can read
Uh,
more about it. Fine.
okay, okay.
Okay. And followed by that. Okay, we are okay. I hope I explained. Now we can go for processing the structure. To process the structure, okay, just a minute. We have to download the entire files.
Okay.
Can you see the download files?
Yes.
Okay. Either you can display files and one by one you can download. This is the PDB file, what we require, the model. Either they will display here or you can download PDB file.
Okay.
What I will suggest is download everything. Don't leave anything like DPU format, metadata, model report. If you download the
Mm-hmm.
model report, can you see? You will get the entire model details.
Mm, yes.
Are you doing simultaneously?
Yeah. I'm
Okay.
writing, sir. Should I do it now?
No, no, no issues, no issues. Recording will be available whenever you are free. You
Mm,
can do. Right.
okay.
Right. So I got the models. Now, since PyMol is installed, I can go and open the model.
Mm-hmm.
But one kind request, then and there you save all the files and keep in the working directory properly. Working directory I refer to as a proper folders.
Mm-hmm.
Now we have confirmed the protein. See how good the protein looks. Now let us look at the sequence. Is there any breakage in the sequence?
No.
So it starts from 1 and ends by 488.
Mm-hmm.
So our domain got model, correct? So we, ha- we,
It's
ha- we have,
fine if it is not up to 515.
no, let us go back to Uniprot. So Uniprot, whatever defined by Uniprot, it is got, we got that. Let us go to Uniprot. Oh, they're saying 27. Okay, okay. What happened here? No. What is 515 minus 27?
Fine.
Okay, I'm not good in maths. 27. 488, correct?
Okay.
So we got what?
480.
1 to 488, correct?
Mm-hmm.
So instead of starting as 27,
Oh, yeah. Okay. One from one.
our model started with 1.
Mm-hmm.
But it's confirmed that the entire sequence is present.
So if it is like that, this amino acid number will be changed.
Of course, but we can refer to the residual position.
Okay. How to do that?
Just a minute. I'll refer to the residue position. Or there is also another way to rename the positions. I'll update in the
Mm-hmm.
next class how to rename the position.
Okay. Okay.
But don't that case will come when we are checking with the interactions.
Yeah.
Or it's a simple math. Like we can give, if it is 1, we can start with 27.
Okay.
We have to add plus 27. That's it. If I'm taking 151, 150 what it refers to.
Mm-hmm. Plus 27 if you add the number on.
Yeah, number will come. 177 it is.
Mm-hmm.
Like that, we can have a simple math. But even though if you want as it is, we also have methods to do that. But that is not an
Okay.
issue here. Fine. So once we got the sequence, now it's time for us to repeat your docking. Hope you know the docking.
Yes, sir.
Okay. You can
Now
also
we need to. I
go
think,
for
sir, you
type.
can show me once how
Yes,
to
yes.
do it. I can, if any mistakes is there in what I am following, I can correct it.
Fine. For that, it requires half an hour because I will
Okay.
show you different modes of docking: blind docking, site-specific docking. Meanwhile, I'll advise you to install a software called Pyrex. Do you have Pyrex in you with you?
No, sir.
You are having
For
Autodoc
one.
Vina.
Yeah.
Okay. Actually, Autodoc Vina is also installed in Pyrex. See, I tried in
Mm-hmm.
several methods, but out of my uh, Out of my experience, I suggest to my participants, students only this method because it is having several interfaces. Can you see
Mm.
that icon? TYRX.
Okay.
Just a minute, it's loading.
Sir, if there is a link, then can you share it with me so
Yeah,
I
I'll
can.
share in the group. You can save it. Can you see? Pyrex virtual screening tool. It's loading. Can you
Mm-hmm.
see?
Yeah.
What do you mean by virtual screening? Virtual screening refers you can do even thousands of molecules at the same time.
Mm.
You can dock even thousands of molecules at the single click.
Okay.
How many ligands you have docked with?
I have not now for this scheme I have downloaded for 20 molecules.
Okay, now you can expand the selection.
Mm-hmm.
You can look for more inhibitors. You can look for even 100 inhibitors.
Mm-hmm.
100 molecules we can dock in a single sitting.
Mm-hmm.
Can you see Vina Wizard, Autodoc
Yeah.
Vina, Open Babel? Same Autodoc Vina is there. We need a special class for completing the entire thing. Once it is ready, only we can move on to uh, MD simulation.
Okay.
So please install Pyrex. I'll share you the link. Any doubts in today's session?
No, sir. I will do it once more. If I have doubt, I'll be keep posting in the.
Please, please
Uh,
keep posting. Okay. I think you are in Telegram, right? You're in Telegram.
I'm not using Telegram. Uh,
Okay, you can use Telegram or you put in classroom. Any urgency, I think you have access to their WhatsApp or something, correct?
yeah, yeah. I have access to their WhatsApp.
Yeah, actually, Miss Bhavani is very active, so she may communicate to us anytime.
Mm-hmm.
Okay, you can check with her. And definitely, I will also check the workspace. Meanwhile, you inform them you have posted something, so I'll go and check again.
Yeah, yeah.
Okay. To find the answer, there is a thing called SourceForge, like where to download Pyrex. Just you click Pyrex download. I'll share the link in the group. Is that fine for you?
Yeah.
It's just few MBs only. You can download easily.
Mm-hmm.
Have you found in chat?
Yes, sir. I will just copy it. Should I do it now?
Yeah, you can do. It's quite
Mm-hmm.
easy only. I think all the software installation you can do after the class. That will be if we can save some quality time.
Mm-hmm.
Like next class, I think next class will be on Monday, right?
Yes, sir.
Will you be carrying your laptop to home, like to India, or leaving here?
Yes, yes. Yes, yes. I will take.
Okay, so you'll have a lot of time to do. Like you install all
Mm-hmm.
the softwares, Pyrex. Sorry, Pyrex you have to install. You have to install. I'll give a list of softwares then to install. Get it ready.
Okay.
You have to install Chimera. Have you heard of Chimera? Do you have in your
Sir,
laptop?
I think I have my camera in my laptop. I have.
Good, Chimera is there. Do you have Discovery Studio? I've seen the images. Discovery Studio is there.
Yes, yes. I it's a
Good. Do you have Ligplot?
like plot. That is online, sir.
No, 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 no. Actually, Lig Plus is online. I'm talking about Ligplot.
Um, no, no. Then I don't have it.
These are very trusted sources. This kind of information, but it requires academic mail ID.
Okay.
You are an institution, right? You are working in an institution. Do you have an
Yeah.
academic mail?
Ye- Yes, sir. I have.
Okay, this is from European Bioinformatics Institute. Please open
Mm-hmm.
that.
Like plot. Okay. Email address I have to enter.
Yes, that you have to register for it. Fine. Same way, I will
Okay.
suggest you because in this 1 week time, whenever you have free time, please keep it ready.
Mm-hmm. I will do it, sir.
Yeah. Same way, there is another thing called VMD. Have you heard of VMD?
I'm having that, sir.
Good. You have VMD. That's great. Okay. Do you have QT Grace? QT Grace. Have you heard of QT Grace?
No, sir, I don't have that.
QT Grace is to, again, view the plots. I'll share that link.
Mhm.
So all the links, please keep it bookmarked or somewhere you store it.
Yeah, I'm copy and pasting in text pad, sir. So keep
Good,
it.
good, good, good. QT Grace, VMD, link plot, and particularly there is another software called ZZ Vega, Vega ZZ.
Okay.
This is again a interesting tool. So I'll make you the best out of your manuscript, but
Okay.
sadly we have a less time. Like we have to make best use of it. Can you see Vegas? That's it.
Yeah.
Since you are from a great university, like do you have an access to quantum mechanical calculations like Homo, Lumo?
No, sir, here it is not there.
Not there. Huh?
Where I'm working, no, no, it's basic. It is not a research center. Only
Ah,
basic research is going. It's a college,
it's a
actually.
college. Right, right,
But,
right.
yeah, so there's no research center. Where I was studying for my MPharm, I was studying in Amrita.
Oh, Kerala, Jaipur,
Yeah, B Farm and MPharm from Amrita I took.
Amritapuri.
Yeah,
Yeah, great, great.
Amrita from Kerala.
You're Kerala,
Hmm?
right? Malayali,
Yeah,
right?
from Kerala. Yeah,
Oh,
from
great, great,
Ernakulam I took my. So
great.
there everything was there, but here I don't. Here there is. It's just a college, so not a
Nice,
research
nice.
institute.
Actually, my boss from ICMR is a Kerala. Like her name is Susan Thomas. You can browse her name. Susan,
Uh
you
huh.
type Susan Thomas ICMR.
Wait one second.
Why I'm saying that, you can refer to the papers.
Okay.
I am very fortunate I have to work with ma'am Susan Thomas, ICMR. You type ICMR.
Susan Thomas. ICMR.
Susan Edicula Thomas. Can you get her name?
Yes, 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 yes.
Ma'am is scientist F, senior scientist.
Uh huh. Okay.
So we were part of it. Like actually, my name is removed now because I just shifted back to Tamil Nadu.
Uh huh.
Here you can look at the research papers, how important
Uh huh.
research is doing, and machine learning methods. So I'm well versed in sequence analysis, database creation, machine learning, disease informatics, protein-protein interactions. These are
Mm-hmm.
my study area, right?
Nice, sir.
So
I can take help from you in future too.
yeah, fine, fine.
Neighborman has given me a very good reference now.
Of course, of course. We see the scientific network is so short. Like we can easily get connected with people, but provided your research interest and my research interest should match.
Yeah.
If it is matching, we can easily have the frequency. At least that should not be any communication problem. Like whatever I'm
Yes.
intended, it's not about the language. I'm saying about the
Mm-hmm.
intention. Whatever intended,
Yes.
you are getting correctly. Right?
Mm-hmm.
And I'm happy that you are finishing the assignment on time. That is very, very important. Since
Uh-huh.
you have
Okay.
shown there is no experimental structures, then we moved on to modeling to
Yeah.
it. The
Mm-hmm.
same thing should be that. So next class, be ready with all the softwares. And my dear Miss Kemi, like it's very, very important to have Ubuntu with you.
Uh-huh. Okay, sir. I will just uh when he comes, I will there is anything that link, WSL link, can you send, sir,
I
once
sent
more?
you this WSL link, first link. I have to send again. I think you stored it. First one.
Okay, that
HTTPS
bookmarked one.
bookmark one, correct.
Okay.
You can even save that like this one.
Okay, so that I have to download and all these softwares.
You
That's it.
follow this. See, actually, they are giving different methods. First of all, you confirm which Windows you are. Windows 10 or Windows 11.
Mm-mm. It was written to Windows 10, sir. And about yesterday we looked, right? It's 10 itself.
Correct. So it's Windows 10. We have to install in two ways. Like we told now, we have to give wwd. We have to install the distributions.
Mm-hmm.
Like, can you see like this distribution names, everything we have to shortlist. Like something will be missing, some additional resources. You can have to browse through, or you have a number of YouTube videos also. In YouTube, you can
Now,
find
if you convert into Windows 11, it is easy thing, right, sir? To convert into Windows
yeah.
11.
Then you have to browse, or you have whatever you downloaded. It is called ISO file, ISO image.
Mm-hmm.
If he is good in dual booting, you can also boot.
Okay. I'm not
Or
familiar with these words, so.
like dual booting means, see, once you switch on the computer, you are going directly into Windows, correct?
Mm.
But in that case, they will ask for option which OS you want to prefer,
Okay.
either Windows or Ubuntu.
Mm-hmm.
So your system have two partitions. The hard disk will be divided. One part will be Windows, one part will be Ubuntu.
Okay.
That's what I'm using for a long time. But later on, going to big institutes, I found there are a lot of smart ways. One such smart way is WSL,
Okay.
where inside the Ubuntu itself, inside Windows itself, without dual booting itself, I can use a small partition. Just you will find an
Okay.
icon Ubuntu. You won't.
Okay.
I have just made everyone to work on this. Nearly 3 to 3 batches have passed through for CAD programming.
Mm-hmm.
More than 20 to 30 people in a single batch. All were able to do modeling and docking within 15 days time span.
Uh-huh. Okay.
But since we are working in your particular protein, we are taking some time.
Mm-hmm.
If it's a generalized class, I can keep on going.
Mm-mm.
So
Okay.
this input is very important because look at here. I'm just typing GMX, Gromax. This is the actual file. We will perform all the experiments.
Mm-hmm.
Gromax MD. Clear?
Okay.
Okay. So thank you so much. And you also one more final touch. You also can browse my profile so that you go through my papers, research articles.
Okay, sir.
The way you have to find my name is Chandrasekhar Ponnuswamy plus ICMR. Can you
Mm-hmm.
see?
Okay, sir.
Now I move to
Yeah.
Ratnam Group tour, but this if you find my name, Dr. Chandrasekhar Ponnuswamy, I'm from the same lab. Oh, this is not me, I guess. ICMR NIRH. You type like this. Type in Google. Is it visible?
Yes, sir.
Share that link also so that I can save.
Yeah. You can share the link, sir. That is.
Yeah. In that you can understand what are the different things. You can see Google Scholar. My name will be like project scientist, comma ICMR. Okay.
Mm-hmm.
In that, I'll prefer you to go for this particular paper called Oscillating Unraveling. In 2021, I published a paper.
Mm-hmm.
paper. There I discuss about each and everything. Fine.
Okay.
Yeah, I'll share this link. Okay, thank you so much. Any doubts?
No, sir. When I perform, if I have any doubt, I'll, as I
Please.
told, I'll keep posting it.
Sure. And kindly save these links and all. Miss Bhavani, are you there?
Yes, doctor.
Yeah, we are done with the class. So any
Yeah.
information to be passed on?
Yeah, sure.